lecture, we started with the female reproductive organ of a flower, that is carpel or pistil. The three parts of a pistil we have seen: stigma, style and ovary. Inside the ovary, we saw a structure which is called ovule, which is called Megasporangium. So that we will continue in today's class, right? So we need to talk about Megasporangium. I told you that Megasporangium is technically called as ovule. It is called as ovule. We described the structure of ovule also. So the ovule is covered by integuments, right? So ovule is covered by one or two or three protective coverings called integuments. Body of the ovule. Body of the ovule is called nucellus, which is a nutritive tissue. The place where the nucellus is absent is called micropyle. Body where integuments are absent. In the body where integuments are not enclosed, that end is called micropyle. There is a pore there called micropyle. The end is called micropylar end. I told you micropylar end represents the apex. Opposite end to the micropylar end is callosal end. Opposite end to micropylar region. What is it called, children? Callosal end. So callosal end represents the basal part of the ovule. The stalk of the ovule is called funicle. All this I explained in the previous class. We are just writing down once. The stalk of the ovule, what is it called? Is funicle. The body is attached to the stalk through hilum. Body is attached to the stalk. What is the stalk called? Funicle. Body is attached to funicle through hilum. Through hilum. I told you this point came in NEET 2020 UG question paper, right? Inside the nucellus is embryo sac. Inside the nucellus lies embryo sac. What is embryo sac called? It's called female gametophyte. Embryo sac is called female gametophyte. Female gametophyte means it is haploid. Why is it female? Because in that egg, the female gamete is seen. So children, this is the description for the ovule. Now, I told you that we can divide the ovule in three types, right? Types of ovules is our next concept. Types of ovules. We can divide the ovules basing on the amount of nucellus they have. On the basis of amount of nucellus. On the basis of amount of nucellus, we can divide ovules into two types. One is called as tenunucellate. Tenunucellate ovules, and the second one is called as crassinucellate ovules. Crassinucellate ovules. What do you mean by tenunucellate ovules? Means in tenunucellate ovules, the development of nucellus is very poor. It is very less. We can tell less nucellus is there. Poorly developed nucellus. Little amount of nucellus or poorly developed nucellus is there, that is called as tenunucellate ovules. Example for tenunucellate is gamopetale members. The flowers in which the petals are fused are called gamopetale members. In gamopetale members, we find tenunucellate. Crassinucellate, opposite, it is massive nucellus, abundant nucellus. Massive nucellus is called as crassinucellate. Then if it is gamopetale here, it is polypetale. Polypetale members. And children, the monocots also. Monocots also have crassinucellate ovules. We can divide the ovules on three bases. The first basis is amount of nucellus. On the basis of amount of nucellus, we have divided them into tenunucellate and crassinucellate. Second one is on the basis of integuments they are having. We can tell the ovule can have one integument, can have two integuments, can have three integuments, can have no integuments. That classification also let us see now. So take a screenshot. Right. So now we will see the second classification also. The first classification is the amount. So the first classification, what we have seen, which is based on nucellus amount, right? And what are the two types we have seen here? The two types are tenunucellate, one condition, and the second one is crassinucellate. So we have seen that tenunucellate means less nucellus, right? Crassinucellate means more nucellus. And we have also seen the examples for tenunucellate. Examples for tenunucellate are gamopetale members and crassinucellate are polypetale members. So examples are gamopetale. The flowers in which petals are fused are called gamopetale. Crassinucellate, let us take the example of polypetale. Means when the petals are free, like hibiscus and all, that's polypetale. With monocots also. Children, this is one classification of ovules. The second classification will be based on, we'll do the second classification also. The second classification, it is based on the number of integuments covering the nucellus, based on the number of integuments. So based on the number of integuments, we can tell no integument, that is ategmic, one integuments, two integuments, three integuments, like that we can go. So if we see ategmic condition, ategmic means no integuments. No integument means the nucellus is naked. Examples for ategmic condition is loranthus is one example. Sandalwood, xanthalum is another example, and Berenophora is another example. Let us write down ategmic. Children, ategmic means how do you expect the ovule to be? This is the nucellus and this is the funicle. No coverings around. So examples are, one is loranthus, other one I told xanthalum, and the third one is Berenophora. These are examples for ategmic. Now, if it is having one integument covering the nucellus, it is called Uni Technique. So the second one will be Uni Technique. So Uni Technique means one integument. So for one integument, what can we give the examples? For one integument, Helianthus and Datura are the examples. How the ovule will be then? One integument means this is the nucellus, covered by only one integument. This is uni-integumented. Examples I told: Helianthus, sunflower. Datura are the examples for unitegmic. Next one is bitegmic. Bitegmic means two integuments around the nucellus. Two integuments around the nucellus. Then two integuments. How do we draw two integuments? It is a common type of ovule. So this is one integument, the other integument. Two integuments. This is what we draw. We saw in the NCERT also. This is the common type of angiosperm ovule. It is the common type of angiosperm ovule. Now, let us see with three integuments is also there. So, the fourth one is tritegmic. How many integuments then, children? It is three integuments. So, tri-integuments, if we have to draw, let us draw the picture, how it will be. So, one integument here we draw. Second integument also we showed. Third integument. This is called tritegmic. Example for tritegmic is asphodilus. Asphodilus is an example. So, we are classifying the ovules on the basis of amount of nucellus, first classification. Second classification is on the basis of number of integuments covering them with examples. Take a screenshot, then we will see the classification of the ovule basing on the positions of the micropyle, callosal end and the funicle. Take a screenshot. Right. Now, we will see the third classification of ovules also. And that classification is based on the position. Yeah, here we go. So, the third classification is based on the positions of micropyle, callosal end and funicle. How are the funicle, micropyle, callosal end are positioned to each other? On that basis, we can divide them into 6 types. The first type is called Orthotrophos. What's the first type of ovule? It is called Orthotropus ovule. Orthotropus ovule, will draw the picture, then we will try to understand, right? So, this is the nucellus and the micropyle apex end is here, right? So, what is this? If we have to draw two integuments, this is the second integument. So, this is nucellus, this is micropyle, this is funicle, right? This is the stalk. The stalk is funicle and the base is callosal end and this end is called micropylar end. If all three are on the same plane, and inside the nucellus we know it has an embryo sac, 3 cells and 2 polar nuclei. So, if all three are on the same plane, then the rotation is 0. So, what is the rotation for Orthotropus here? 0 degrees angle. Like all three are on the straight line. All three are on common axis. Such a type of ovule is called as Orthotropus ovule. Example for Orthotropus is Piperaceae family members. Orthotropus, what is the example? Piperaceae family members. Now, let us go with the second type of ovule. In the second type of ovule, if we tell it is Hemianatropus. in Hemianatropus, rotate the Orthotropus ovule to 90 degrees. So when you rotate it to 90 degrees, the nucellus, which is straight, it becomes like this. And then the integuments go on this way. Right? So, and the embryo sac is inside here. 3 cells here, 3 cells here, and polar nuclei. So this, is it showing 90 degrees from the funicle? The micropyle is at right angles to the funicle. So what should we write here? The micropyle is at right angles to funicle. The micropyle is at right angles to funicle. And how much degrees rotation? 90 degrees rotation. Then what is it called? It is called as Hemianatropus. Example for Hemianatropus is primula and ranunculus. What example shall we write here for Hemianatropus? Primula, one plant. Ranunculus is another plant. Now let's go with the third type of ovule. The third type of ovule, which is a common type in angiosperms, is called Tropus Ovule. Here it is straight. Here you have made a rotation of 90 degrees. Now bend it to 180 degrees, right? So in Anatropus, make one more 90 degrees, means it will rotate this way. Then this is one integument and this will be the other integument. This will be the other integument, and here we have the embryo sac. These 3 cells here, 3 cells here, and a gap. Those two are the polar nuclei. Now what is this? In this, the micropyle is near to funicle, right? So this is Anatropus. Rotation is 180 degrees, as you have seen. We have rotated complete 180 degrees, and then we are telling that the micropyle is near to, this is micropyle and this is funicle, near to funicle. Then it is called Anatropus. This is a common type of ovule for angiosperms. We can tell this is common type in where? In angiosperms. Any doubt still here? You have seen the first type, Orthotropus. Gymnosperms have Orthotropus ovule, right? And next one is Hemianatropus, primula and ranunculus. Third one is Anatropus ovule. For Anatropus ovule, most of the angiosperms will have. Next one will go with Campylotropus. The fourth type of ovule is Campylotropus. Now, in Campylotropus, what we will do is, it is Hemianatropus only. We will rotate it little bit, like curvature will be there. I rotated the nucellus. So the body will rotate. The body is rotating, whereas the embryo sac will not rotate. The embryo sac will be normal only. You can see there's a curvature in the body. Then that is called as Campylotropus. For Campylotropus, we can tell the rotation is 90 to 160 degrees rotation, because it's not complete 180. From 90 to 160 it rotates. So there is curvature in the body of the ovule. You can see there's a curvature in the body of the ovule. Example for Campylotropus is Brassicaceae members and Fabaceae members. What are the examples? Brassicaceae and Fabaceae members. Now coming to the next type, it is called as Amphitropus. In Campylotropus, only body has showed the curvature. In Amphitropus, body also and the embryo sac also will show the curvature. Body showed a curvature like a horseshoe. This we know, we drawed it for Campylotropus. But in Campylotropus, we told the embryo sac is normal. Here in Amphitropus, even I have to rotate the embryo sac also like a horseshoe. Embryo sac is also having horseshoe type of curvature. 3 cells here, 3 cells here. This is called as Amphitropus. Example for Amphitropus. Now the rotation for Amphitropus is complete turning, right? 160 degrees we can give. Example we can take it as papaver as an example for Amphitropus. What example shall we write here? Papaver is one example we can write, and Butomus is another example. So last type of ovule is called Sarcinotrophosis type of ovule. What's the last one? Sarcinotropus ovule. It is 180 degrees means half the way. 360 degrees rotation is Sarcinotrophosis. In Sarcinotrophosis, how much rotation? 360 degrees angle it will rotate. Now we have to show the rotation. See, this is funicle and the funicle has made a 360 degrees rotation. So the embryo sac will be here. This 360 degrees rotation ovule is called Sarcinotrophosis, is found in Opentia desert members. It is found in Opentia. So children, we have seen the types of ovules on the basis of the nucellar content, tuninicellate and crassinucellate, on the basis of number of integuments, ategmic, unitegmic, bitegmic, tritegmic, on the basis of their relative positions, on the basis of where is micropyle located with the funicle and color is this classification. If all are on the same axis with 0 degrees angle, Orthotropus, Pipraceae example. If it is making a 90 degrees angle, then it is Hemianatropus, Primula, Ranunculus are the examples. If it is 90 here, now it has made 180 degrees rotation, then that is called Anatropus, which is a common type of angiospermic ovule. Next one is Campylotropus. In Campylotropus, the body curves, whereas the embryo sac will be normal. Rotation 90 to 160 degrees rotation. Example, Brassicaceae and Fabaceae. Next one is Amphitropus. Body also will curve, embryo sac also will curve. Example, Papaver and Butomus. The last one, complete 360 degrees rotation. It is Sarcinotrophosis. Example is Opentia, 360 degrees rotation. So with this, we completed types of ovules. In the next lecture, we'll talk about megasporogenesis and the embryo sac formation. If you like the lectures, like, share and subscribe to my channel. Thank you.